In this video, we'll be looking at some of the last topics in this chapter. So we're going to pick up where we left the last one. We're about to talk about neuromodulators. And neuromodulators are chemicals secreted by neurons and that can have an effect on a group of neurons or individual neurons. And it can either enhance their action or possibly inhibit their action. And so it may alter the rate the neurotransmitters are synthesized, or the rate at which they're released, or how much is released, their reuptake, or their breakdown. So there's a number of mechanisms within this group. The other thing that they may do is they may adjust the sensitivity of the postsynaptic neuron. So let's have a look at some examples. Nitric oxide is a sample neuromodulator, and it enters the postsynaptic cell and activates a second messenger pathway. Neuropeptides, of course, look at the name, neuropeptides. They're a chain of amino acids that can act. Enkephalins, endorphins, which we quickly had a look at when we're looking at the chemical classes of neurochemicals in the last video. They can actually inhibit pain signals in the CNS, and that's something we'll touch on in future chapters. Now, we're going to talk a bit about neural integration, and neural integration is basically the ability to process, store, recall information, and this is really at the heart of how we make decisions, either consciously or unconsciously, for certain things to happen in the body. And just a reminder that brain cells are very well connected, allowing for real complex integration. For an example, one group of cells, the pyramidal cells or pyramidal cells, you can say it either way, of the cerebral cortex have about 40,000 contacts with other neurons, and that's a mind-boggling number. So there's a lot of integration that can occur there. Now, the trade-off is chemical transmissions involve a synaptic delay that makes the information travel slower than if there was no synapse. And we talked about that a little bit in the previous video. That, yes, if you have to release neurotransmitters from a presynaptic neuron to a postsynaptic neuron and have them bind to a ligand gate and then cause their action to occur, then that's going to eat up a little bit of time. Now, it's a very small amount of time, but you gain a real advantage. And that is, it allows the neurons to do better decision making. So you're going to see things over the next few minutes. We're going to talk about things that we've talked about in class. And in class, when we looked at excitatory and inhibitory neurons and their effects on dendrites and how they may impact the trigger zone in the axon. That is really what we're about to talk about here, but we're going to introduce some new names in that we're going to start talking about postsynaptic potentials. And we're going to look at two types: excitatory postsynaptic potentials and inhibitory. Now, just think of the names. Excitatory potentials are those potentials that basically cause the inside of the cell membrane on the cell that's receiving the signal to become more positive and get you closer to that magic point of threshold. And not surprisingly, inhibitory ones that we're talking down here, they're the ones that will actually hyperpolarize. And we had a look in our class discussions about how an excitatory presynaptic neuron may release neurotransmitters that could open something like a sodium gate, and sodium will flow into the postsynaptic neuron and hopefully increase the voltage in the trigger zone and get you closer to threshold. Whereas we also looked at inhibitory, and in that case we looked at opening of, say, chloride gates. And because of their negative ion, it makes it less likely, so you hyperpolarize. And really, this is what we had talked about. We just didn't give it the names of excitatory and inhibitory. So when we're looking at postsynaptic potentials, different neurotransmitters can cause different types of postsynaptic potentials. For example, glutamate and aspartate can produce excitatory postsynaptic potentials. That's what this stands for, EPSP, in the brain. Okay? Whereas glycine and GABA can produce inhibitory. So one of the things that dictates whether you're going to have an excitatory or inhibitory effect on the basically on the neuron beyond the synapse, aka the postsynaptic neuron, is the neurotransmitter. But that being said, and we mentioned this once again in previous video, is that sometimes it's also the receptor that will determine the effect that is carried out. And so a neurotransmitter might excite some cells and inhibit others depending on the receptor type in the postsynaptic membrane. Now, also, as we looked at in class, we really talked a bit about how it's not the most common thing is where one neuron interacts with one neuron. There are many parts in the body where that happens, that one neuron talks to one neuron and then it passes a message on to the other one, kind of like the game of telephone that kids often play. Now, in this case, one neuron can actually receive input from thousands of other neurons. And as we said in our in-class discussions, sometimes they're excitatory and sometimes they're inhibitory. And a neuron's response depends on whether the difference between the excitatory and inhibitory, gets you to basically threshold. And of course that process is called summation, where I add up all the possible influences from excitatory and inhibitory. And of course what we're trying to do is influence the trigger zone. So really, it's a balance of the excitatory postsynaptic potentials, remember that's what this stands for, and inhibitory postsynaptic potentials that allow the nervous system to make decisions. Now, this hopefully seems familiar because this is what we talked about in class, but let's go over it one more time. There's two types of summation. I can have temporal summation, and that's when one single synapse generates excitatory postsynaptic potential so quickly. So one neuron keeps firing and sending across neurotransmitters to the postsynaptic neuron. And of course, this can give you an additive effect. And because it's based over time and increased frequency, we call it temporal. The other example we looked at was spatial summation. And spatial summation is when a number of neurons are trying to talk to one postsynaptic neuron. And in this case, the overall effect as to whether a trigger zone will hit threshold is based upon a number of neurons talking to that one neuron. And because those neurons are synapsing at different parts in the dendrites or even in the cell bodies, this effect is coming from a number of locations on that postsynaptic neuron. It's getting influenced, so we call it spatial summation. So we get simultaneous input from multiple presynaptic neurons on the postsynaptic neuron. Now, here's just sort of a diagram of it, just to make sure everyone sort of has a clear understanding. Here's the postsynaptic, because there's our synapse right there, so that's the postsynaptic neuron. These are presynaptic neurons, because of course they're before the synapse. Okay. And here we have a case, a very simple model, but it illustrates the point, is that here there's a number of neurons that are trying to talk to this one. Now remember, this neuron right here, its trigger zone has to hit threshold, and that basically means things like sodium ions have to flow down there. And as we said in class, one of the ways we can do that is I can accomplish that if one neuron keeps firing. So it fires once, fires again, fires, keeps sending action potentials one after another down its axon, and that releases a whole lot of neurotransmitters over a short period of time. And that should keep things like sodium gates open long enough that the sodium will diffuse to the trigger zone, and then this cell will go ahead and fire. So that is a type of temporal summation, because there's a time period that the number of firings are spread over a very short period of time, and temporal refers to time. The other case is a spatial summation. In this case, what we have is, once again, we have to influence this trigger zone to hit threshold for a firing of the postsynaptic neuron. And in this case, what we do is we have a number of neurons working together, and they will send signals down. And because each one of these is influencing a different part of the neuron, and the cumulative effect of, say, those sodium ions flowing down here will cause the trigger zone to fire, it's spatial summation because it's spread over a space. But the end result is the same. We have to get the trigger zone of the postsynaptic neuron to hit threshold, and when it hits threshold, then the action potential will occur. And here we can see it graphically, that here we're at resting membrane potential. And, say sodium gates open, and then they close. Well, sodium gates open again, and then they close. Sodium gates open again in the dendrites of the postsynaptic. And we're starting to get that influx of sodium into the cell. And if I can get enough sodium to diffuse down to that trigger zone, I hit that magic point, which is known as threshold, and that's when the voltage regulated gates in the trigger zone open. And of course, then we have an influx of sodium as sodium channels open, gated channels open, they're voltage gated in the trigger zone. They start to close, and then we have basically potassium gates open, and potassium now rushes out of that part of the membrane of the postsynaptic. And as we mentioned, you actually overshoot. And when you overshoot, you end up with a little hyperpolarization here, which we talked about in previous videos. And this is basically an action potential. And all we're showing you here is that in order to get threshold, a number of excitatory postsynaptic potentials can add up through summation, either temporal or spatial summation, to get you to threshold, and you get your action potential. Now, when it comes to presynaptic facilitation occurs when one presynaptic neuron enhances another one. Okay, let's have a look. That we can also do. Now we had just finished. Just to back up for a second, we just talked about facilitation. Summation, I should say. We finished summation. We're about to talk about facilitation, and this is where one neuron can kind of direct to aid a neuron in firing. So it increases the likelihood of a synaptic transmission. And let's have a look. Here's one example. Here's a model we can look at of presynaptic facilitation. In this case, you notice that we're actually down. Here's the axon right here. We're down near the bottom of the axon. And this neuron, because as mentioned way back, not all neurons interact with the postsynaptic neuron in the dendrites or the cell body. Some of them actually come in and basically interact with the axon. And in this case, this one is designed to facilitate the firing. And if this neuron does not fire, there's no activity in the facilitating neuron. Neurotransmitters released here, and I get very minimal activity. And if there's no activity, very little neurotransmitter released, that's really going to decrease the chance at the postsynaptic. So these types of neurons, they interact often down near the bottom of the axon or somewhere along the axon. And they're sort of trying to influence, despite what may have happened up in the cell body, whether the cell will fire or not fire. And in this case, if it's a facilitating neuron and this neuron does kick in and go, hey, despite whatever, I do not want you to actually release your neurotransmitters, even though you may have got a signal up high. I'm going to kind of override it at the last minute. Or I could enhance it. And in this case, it's a facilitating neuron, so it's going to enhance it. So if this one fires, what happens is a neurotransmitter is released, and the neurotransmitter comes into here, and it has a biological effect, which is makes it much more likely that neurotransmitters will be released. So I get an enhanced release of neurotransmitters. And if I have extra neurotransmitters released here, then I'm more likely to have a postsynaptic effect. Now, so that was sort of encouraging a neuron to fire, or to fire more, or to fire less. Now, we also have presynaptic inhibition, and this is when one presynaptic neuron suppresses the other. So the case we just looked at was the case of facilitation. Facilitation means making it happening easier. This is a case of inhibition. And what's the goal of this? It's, well, despite a postsynaptic neuron trying to fire, and a neuron's going to fire, at the last minute, this kind of jumps in and goes, hey, really reduce the amount of neurotransmitters you're releasing. I want to suppress the effect that you're having. So it reduces or halts unwanted synaptic transmissions. One example is, inhibiting neurons may release something like GABA, which prevents the voltage-gated calcium channels from opening. So little or no neurotransmitter is released. And let's have an effect. Let's have a look. So this diagram looks similar to the one we just looked at, but the difference is that the first time we looked at this a few slides ago, this was a facilitating neuron. So when it released neurotransmitters, it was basically saying, hey, if you got a signal to fire, I want you to release a whole lot of neurotransmitter. In this case, this is presynaptic inhibition, where an inhibitory neuron down this area, it's saying to itself, you know what, I do not want this neuron to fire. Or if it fires, I don't want much release of neurotransmitters. So if the inhibitory neuron's not firing, the action potential comes down, and if there's no inhibitory effect at the last minute, then it's going to release neurotransmitters and carry on like it usually does. But let's take the case over here. Then in this case, if this neuron has an action potential and it's getting close, it's sweeping down towards here where the neurotransmitters are, the inhibitory one kicks in at the last minute. So this one's kind of past all its checkpoints, is that, up in the trigger zone, it did have a depolarization, the action potential sweeping down, and this inhibitory neuron right at the last minute kind of jumps in. And as we said, it could be the case of, say, GABA, and GABA causes an influx of basically negative ions. Okay? And of course, if you hyperpolarize this area and the action potential comes down, you may basically keep things like the calcium gates from opening. So, in the first case, the calcium gates action potential came down. I had a local depolarization here. Calcium gates opened, and calcium came in, and that's the signal for the release of neurotransmitters. If the inhibitory neuron kicks in, the action potential comes down. And when it gets down here, the inhibitory, as I said, the inhibitory one has released neurotransmitters that have made the inside here more negative. They've hyperpolarized it. So when the action potential comes down, this area is very negative. And that basically negates the action potential, and this voltage-gated calcium channel cannot open. And if that calcium channel doesn't open, it's the calcium that floods in that is largely responsible for these neurotransmitters to get released, and I basically shut down. So this thing about facilitation and inhibition are ways at the last minute to say, you know what, axon, yeah, you think you're going to fire, you have an action potential coming down, I'm going to stop you right down near the synapse and override it. Now, we're going to end the video there, and we're going to come back and talk about neural coding in the next one.